In this video, we will discuss the principles of photometric enzyme assays. To understand enzyme reactions, we need a reliable and reproducible method to measure enzyme activity. An enzyme assay is a procedure that allows us to measure the activity of an enzyme under specific conditions. Photometric assays are of particular importance in biochemistry. In many cases, we can use such assays to follow enzyme reactions in real time. Photometric assays can be used when the substrate, product, or cofactor involved in an enzyme reaction absorbs light. These assays are widely used to study enzyme kinetics, such as reaction rates or substrate conversion. To perform photometric assays, we typically use an instrument called a photometer. A photometer has a light source that emits a light beam. Usually, this light is in the range of 200 to 800 nanometers. Initially, the light passes through a monochromator. This component isolates a specific wavelength from the light emitted by the light source. The selected light then travels through a cuvette containing the sample. The cuvette is placed in a sample holder. Finally, a detector measures the amount of light that passes through the cuvette. A so-called chromophore, a light-absorbing molecule, has to be consumed, produced, or modified during the enzymatic reaction. Changes in its concentration due to enzymatic reactions result in changes in light absorption. These changes can be used to quantify enzyme activity. To understand how photometric assays work, it's important to understand some fundamental terms and their relationships. The key principle behind photometric assays is the Beer-Lambert law. This law states that the absorbance of a chromophore is directly proportional to its concentration. In this equation, A represents the absorbance or the amount of light absorbed by the sample. Epsilon, the molar absorption coefficient, is unique for each chromophore. It is given in units like per molar per centimeter. This coefficient links the absorbance of a molecule to its concentration. It provides a relationship between how much light is absorbed and how much of the molecule is present in the solution. C is the concentration of the chromophore, expressed in mole per liter. L is the path length, which is the distance the light travels through the sample. For most measurements, L is 1 centimeter, which is the length of a standard cuvette. By understanding these components, we can now calculate and interpret absorbance data. Let's consider an example. Here we see the spectrum of the molecule paranitrophenol. It appears bright yellow at an alkaline pH of 9. When analyzed with a photometer, paranitrophenol shows maximal absorbance at a wavelength of 405 nanometer. In this case, we have a paranitrophenol solution of an unknown concentration. If we put this solution in a photometer using a standard cuvette, we measure an absorbance of 1.16. We know the molar absorption coefficient of paranitrophenol, which is 18.3 per molar per centimeter. By applying these values to the Beer-Lambert law, we can now calculate the concentration C of the solution. Paranitrophenol is widely used in enzyme assays because it can be attached to various groups like phosphates, acetate, or palmitate. Here, an esterase is mixed with the substrate paranitrophenol butyrate. This molecule is colorless. However, if the esterase is active, it releases the paranitrophenol group, which strongly absorbs light at 405 nanometer. This property allows us to monitor enzyme activity. The higher the activity of the enzyme, the more paranitrophenol will be released. Thus, the higher the absorbance at 405 nanometer. However, it is important to understand that the absorbance of chromophores can vary under different conditions, particularly with pH. Shown here is the pH dependence of the paranitrophenol absorbance. Note that for all spectra, the same concentration of paranitrophenol was used. At alkaline pH, such as 10, paranitrophenol is fully deprotonated, resulting in maximal absorbance. When we lower the pH toward acidic pH, paranitrophenol becomes protonated. its absorbance at 405 nanometer decreases significantly. This highlights the importance of controlling the pH during the measurements. To achieve reproducible results at 405 nanometer, we need to make sure pyronitrophenol is fully protonated. Alternatively, we can utilize the isospastic point of pyronitrophenol at the wavelength of 347 nanometer. An isospastic point is a wavelength where the absorbance remains independent of pH changes. This wavelength can be used to measure esterase activity independent of the pH value. However, the signal output will be lower than at 405 nanometer. Let's take a closer look at the enzyme reaction from before. When we studied the esterase reaction, we mixed the enzyme solution with the substrate pyronitrophenol butyrate in a suitable buffer solution. In this experiment, the reaction buffer has a pH of 9 and the total reaction volume is 1 milliliter. When we analyze this reaction in a photometer and measure continuously at the wavelength of 405 nanometer, a progress curve can be observed. This means that the enzyme is actively converting the substrate. We can follow the steady release and accumulation of pyronitrophenol over time, which absorbs strongly at 405 nanometer. The slope of the curve, given as delta absorbance over time, is a direct measure of enzyme activity. The steeper the slope, the higher the activity. Importantly, when using such progress curves to calculate enzyme activity, the product formation over time must increase linearly. If the curve deviates from linearity, the enzyme concentration in the assay could be too high, or the substrate concentration may be insufficient. To ensure reliable results, always include blank measurements that do not contain the enzyme to account for non-enzymatic background signals. How can we calculate the enzyme activity from such a progress curve? Let's start with the basics. Enzyme activity is the ability of an enzyme to catalyze a specific reaction. It is defined as the amount of substrate that an enzyme can convert into a product per minute. Enzyme activity is typically expressed as enzyme units U. By definition, 1 unit corresponds to the formation of 1 micromole of product per minute. Be aware that this definition does not apply to restriction enzymes. What we can obtain from a progress curve of an enzyme reaction is the change of absorbance over time. This represents a reaction rate and is usually expressed as absorption units per minute. In order to calculate enzyme activity in the form of units, we need to convert this absorbance change into a change in molar concentration. This can be done by using the Beer-Lambert law, which relates the absorbance of a molecule to its concentration. By rearranging the Beer-Lambert equation, we can solve it for the concentration C. This step allows us to convert the absorbance changes during an enzyme reaction into changes in the molar concentration of the product. By substituting the absorbance with the absorbance change over time, we can calculate the enzyme activity expressed in micromoles per minute. To do this calculation, we need to know the molar absorption coefficient of our product and the path length of the cuvette, which is usually 1 centimeter. Enzyme activity is usually reported in units per milliliter of the reaction, which is called volumetric activity. The volumetric activity is a practical and standardized way to express enzyme activity. It indicates how much enzyme activity is present in a milliliter of the reaction mixture. By incorporating the total reaction volume V into this equation, we can normalize the activity to a standard reaction volume of 1 milliliter. Practically, we can use the following formula when calculating the volumetric activity from absorbance changes. This equation includes the sample volume V, which is the volume of enzyme solution added to the assay. Very often, the enzyme solution has to be diluted because it is too concentrated or too active. In this case, the dilution factor D also needs to be considered. Last, we have to add the molar absorption coefficient, which, for example, is 18.3 per millimolar per centimeter for pyronitrophenol. Knowing this data, we can calculate the volumetric enzyme activity. Another important parameter used to describe enzyme activity is the specific activity. This represents the enzyme activity per milligram of protein, expressed in units per milligram. Specific activity is calculated by dividing the volumetric activity by the protein concentration of the sample. The specific activity is an indicator of the purity and efficiency of an enzyme.